afternoon. It's the Singapore story on COVID-19 because outside of China, in fact, Singapore is one of the earliest countries that have been dealing with COVID, and unfortunately, we are still at it. I think the various teams have been pretty exhausted, but I have to say that with all that has been happening, all the new discoveries, the new technologies, and also networks like what we are enjoying, this moment, I think it will actually help prevent future pandemics. I'm not going to be speaking about SARS CoV-3. Maybe you can ask Julian and his boys what they are. But like I said, I will be sharing with you how actually basic researchers like myself work closely with the clinical community and put our knowledge to use to help the country that also help refine some of the policies. So I will be sharing with you some earlier studies in 2020 when we were dealing with the unknowns. So we were just establishing tools from nothing, and how the journey through the mass vaccination in 2021 up to this point, and the constant battle with new variants while the country has been going through the vaccination process, and it was a little bit of a disaster last year, but I think we have overcome that this year. So I believe some of you might have remembered me from my previous life when I was a PI at the Singapore Immunology Network. So actually over the past 2 years, A-STAR itself has also been reorganizing, and so the A-STAR Infectious Diseases Labs was established actually during the pandemic. So I have to say that the core capabilities is to drive and deliver translational infectious diseases research, and how could we develop a cohesive program on epidemic preparedness that could actually align with national priorities. So obviously, basic research continues to be important, and that is the core capabilities by all the investigators to understand infection and immunity on various pathogens. And you could see from the top right panel are the 3 pillars that are important: antimicrobial resistance, respiratory diseases, not just COVID, but flu and some of the other bacterial infections that are very predominant in Southeast Asia. And also vector-borne diseases, which is actually where my previous life, but I have to say that we are trying to complete some of the previous projects and also start new ones. So I'm not going to go into details here, but for the early career researchers who might want to understand more about the A-STAR Infectious Diseases Labs, please just drop me a note, or we could have a chat during the coffee break, because we are in the midst of recruiting more young investigators. So these are the current research groups, right? So the various teams, and our most recent recruit is Marco Vinuzzi from Pasteur. So he joined us in 1st September. So he would be he has been growing his team locally, and there are also other investigators that have joined us from Australia and also from within A-STAR itself. So working on the various pillars, as I mentioned earlier, from fungal infections, bacteria, and viral. So I will just share with you the COVID story that that has started as early as end January 2020. So I believe at that time, I think many countries were still quite blissful, while in Singapore, it was actually quite close to the Lunar New Year. And there was this a struggle actually with our local authorities, whether should we close our borders, because it's also that time where we have many Chinese tourists that would be leaving the country to come to Singapore for their vacation. But I guess it was a decision that came too late, because it was just 1 day before the Lunar New Year that we had our first index case. So of course, the rest is history, and I think many of the research groups had to pivot our resources and also expertise to play a role in COVID-19. And so right now, I think the term that we use in Singapore is living with COVID, despite the fact that we still have more than 10,000 cases a day. And most of the safety measures have been relaxed. So, so over at A-Star, my research team and various investigators then work closely together with the other people from the local ecosystem. They are mainly from the National Center for Infectious Diseases, which is the main hospital that deals with all the infectious diseases outbreak cases, the National University Health System, and also our collaborators from the Duke NUS. So I believe many of you are also well acquainted with Lin Fa Wang, who has actually made a lot of a significant impact during the COVID-19. So this just gives you a general overview on the approach from basic virology to various areas of molecular immunology to understand immunity, and also the development of various immunoassays that will enable us to understand the signatures, inflammatory signatures, and also various aspects of the mechanisms of the disease progression, immune correlates of severe diseases, and protection. So one of the earliest thing that we had to overcome was to manage how do we bring some of these clinical samples out of the BSL-3 into a BSL-2 plus to enable us to look at to study the cytokines responses. And I have to say that it took 2 months of fight, continuous fight, to get the approvals and also to get them out. But we had to prove, use various inactivation methods to enable us to prove to the Ministry of Health that it is safe to take the samples out. So when that happened, we went straight on to look at the inflammatory responses from the patients. And this was one of the earliest studies that we published in the earlier part of 2020, was the first 100 cases of the COVID-19 patients. So you could see from here that the clinicians classified the patients into 3 categories: the mild, no pneumonia cases, the slightly more severe pneumonia cases but without hypoxia, and the more severe pneumonia cases with hypoxia. And needless to say, the patients on the far right, the most severe cases, were many of them ended up in ICU and also death. But you could see from here, at that time, with this approach, we were able to identify clear signatures, inflammatory mediators in the plasma of these patients, that then allowed some of the clinicians to push ahead with some early days of therapies. So because as we know, this has been, of course, this came out slightly later on, where the mention of a cytokine storm and how the implications of severe diseases that then potentially enable, like I mentioned, the early days preliminary therapies. So obviously, the use of a non-steroid anti-inflammatory drug such as the COX-2 inhibitors have been used for other viral infections before, specifically flu, that has shown to be useful. So this was one of the earlier therapies that the clinicians attempted that were derived from the earlier cytokine responses. And from this small cohort, I will say that success was fairly mixed, but again, the number of patients that they attempted were also fairly small. But you could see from here that the level of IL-6 actually declined after the treatment in about 83% of the patients. Now I have to say that obviously in this small cohort, the more severe cases were also the elderly. But needless to say, it was one of the earlier reports that we mentioned that the IL-6 could be a potential therapeutic target. Now, obviously, this is fairly outdated with the wealth of papers that have been coming out over the past 3 years. So I was just sharing what with limited resources at that time. Now, the next thing that we attempted to work on, as I mentioned in one of the earlier slides, was to look at the immune phenotyping at the cellular response. So this was something that it was also another, I have to say, a challenge, but then a success, and it was actually led by 3 very brave postdocs, that were dabbling around with panels, their fax panels that were looking at other diseases, and they then wanted to push this ahead. Now, then another challenge was how were they going to do this in a BSL-2 condition. So I will keep that story for another day, but we managed to overcome that. So from this group of patients that we have managed to collect, you could see from here that the acute signatures had high levels of CD169 positive monocytes, and also CD11B high neutrophils, which, we would have expected this, but this was one of the earlier studies that we actually showed this from the acute patients. And you could see from here that when the same group of patients recovered, the signature changed. So the take-home message from this particular story was that we showed that while on the left panel, by looking at the neutrophils population, we did not actually observe any significant changes, while we did observe a higher number of total neutrophils, which was expected in any infection. But when we went deeper to look at the immature neutrophils population, and that's where we were able to identify the significant differences that could even be associated with the increasing disease severity. So this was one of the points that then enabled us up to this day to look at immature neutrophils in terms of disease severity, and how we then use it to compare with the other VOCs later on. So, so subsequently in the second half of 2020, when we managed to overcome, to control the widespread of COVID, then there were another group, another cohort that the clinicians got really interested in, which was the asymptomatic patients. Now, this would be people within the same household of the confirmed cases. And I have to say that the classification of our asymptomatic patients were very stringent, where they were totally symptom-free, but PCR positive. Now, there were some other studies that were a bit more relaxed about it, but in this case, we only recruited the PCR positive and asymptomatic cases. And you could see from here that by then, it was really suggested the involvement of T-cell responses. So using the a larger panel here, we could see from here that the asymptomatic patients actually exhibited more robust virus-specific Th17 responses, right? So, which then led us to use all this baseline data when the time of vaccination was rolled out. So the first Pfizer vaccine actually arrived in Singapore in December 2020. So at that time, only the healthcare workers were the first ones to get vaccinated. And so we then started recruiting a group of the vaccinees to actually look at their responses. And subsequently, we then had Moderna vaccine, which we also included. And so I will just move on to a little bit from a different angle, which is the detection of antibodies, which proved to be extremely useful to monitor the vaccine responses in vaccinees. So I think this is just an introduction. Now, I just wanted to share with you while we were using available reagents, but the research groups also tried their best to establish new assays. So one of them was the what we call the S-flow assay, which was developed in the lab of Lohan Rainier, where it's they use the entire S antigen. And with this, you could see from here that the top panel, well, this was just based on the natural infection. The detection was very accurate and sensitive and fairly high throughput and quick. And using that assay, we then continue on to assess the IgG responses in the vaccinees, as I mentioned. And so obviously, for the whole of 2021, then we collected the other vaccinees, and you could see from here that there were also the elderly in the population. But so using the S-flow, we were able to show that actually almost everyone had detectable IgG after. Obviously, there were a low percentage of some individuals that were low responders, but subsequently with the boost, they would also have gotten a very healthy detectable level of IgGs. And obviously, we can't be using just the original isolate, so there are different versions of the S-flow as well that is covering all the variants. So so this was the assay that we have been using quite actively up to this day. So as I mentioned, then when it came to sometime in the middle of maybe around April 2021, then there was the Delta outbreak. And this was a time where Singapore was still in the midst of providing the vaccine, and then there was some resistance in some of the people. So I really liked the earlier talk on social sciences and how you actually convince the people to get vaccinated. I think this was something that Singapore did not anticipate, some of the resistance to get vaccinated, despite the fact that everything was provided. So we were in the midst of going through this process, and we had the Delta, and there were actually a big population of people who were not vaccinated, or people who only had one shot and they got infected, and it became a mess. So then we started collecting people who were vaccinated and yet had the infection again. So it was the question of vaccine breakthrough. So at this time, we had to go deeper to understand more of the B cells at this stage. So just a very, very brief introduction, which I believe many of you are familiar, where when we had the first infection or exposure, then obviously the B cells will then have the first responders of producing the antibodies, right? But then there are also memory B cells, which we like to label them as reservists, because the national service is still compulsory in Singapore. So this was actually prepared by one of the young investigators who had gone through the army. So in his mind, he said, okay, it's a reservist, if you put it that way. And when you have the second encounter, what happens is that you actually rely a lot on more on the reservist, which is the memory B cell, right? It will be much quicker. So so using this concept, we then went on to understand vaccine breakthrough. And if you could just see from this slide, by measuring antibody responses only, right, in the middle, the last panel, the S-flow, you could see that actually there's no significant difference in terms of the levels of antibodies. And if you were to compare with the C-PASS, which was the assay that was developed by Lin Fa Wang, you could also see that it's not significant. And we also had the pseudovirus neutralization, comparing between the wild type and the Delta, also non-significant. So actually this shows very clearly that the vaccine breakthrough cases actually did not have inferior plasma antibody responses. So actually what happens then? What is it that made the difference? So what this study actually showed that while the levels of plasma antibodies had no difference, it was the vaccine breakthrough cases actually had lower memory B cell responses compared to the close contacts, as you could see from here. So then it just confirms the importance of memory B cells in terms of protection. So which sort of verify the earlier hypothesized diagram that I showed you earlier. So in fact, it is the quality of the antibodies from these memory B cells that were more superior. You could see that the delta change was much more significant. So actually, this just means that, you know, because the antibodies from the memory B cells are actually a lot more diverse as expected. So that explains why the efficiency of protection was much better. So I'll just move on to the last part now. You know, okay, this just shows the differences in terms of the inflammatory responses between vaccinated and non-vaccinated people. So the last part of it was when we thought that we were out of the woods by November to December 2021. Then there were the reports of Omicron that was happening in Europe and other parts of the world. So again, we were getting prepared. So by near Christmas 2021, so we had our Omicron cases. And so at that time, by January to early February, we had a decent number of cases already. And but that moment, the clinicians actually clearly indicated in the vaccinated people, the disease was actually fairly mild in comparison. So, you know, based on all the earlier data sets that we had previously generated, we then actually were enable us to make comparisons almost immediately, where we also found that in terms of the systemic cytokine responses, in fact, the breakthrough cases actually had fairly moderated or mild signatures in comparison. Then we went to compare with our cellular data sets, and once again, it sort of verified that actually only small differences were actually found in terms of immune cell dysregulation. And actually there was minimal change in terms of the neutrophil numbers, and also just a slight decline in T cells. So I think with this, it sort of gave a lot of confidence to the clinical community that it's going to be fairly manageable if we can manage the number of cases going into the ICU. So I just want to finish off, you know, that we are not done. Unfortunately, we are still battling, living with COVID. But I just want to share how the support of the research community were able to help guide a lot of the policy changes. And I have to say that it was part of these studies, not just from my lab, but like I mentioned, the whole of Singapore sort of then gave a lot of confidence to the healthcare and also the government that it is okay to not have another lockdown, but enable us to have a more relaxed measures. And it was in stages. You know, every month there will be changes effectively. So I think to the younger researchers here, I just want to sort of show that actually the impact that scientists do actually can be quite great if you were to have a lot of conviction and believe that you will, in whatever contribution that you do, you will make a difference. Thank you for your attention.
Perfectly timed. Thank you. Questions for Lisa?
Very nice presentation.
Thank you.
Just in terms of your vaccine kind of peer responders, were there any risk factors you identified?
Lisa, do you want to repeat the question?
Okay, so you are asking the low responders from the vaccinees, any risk factor? The elderly, yeah, and some of them also were, they had some other kind of immune-related disorders, but it was also for some of this group, they also responded quite badly to the mRNA vaccine. So we actually stopped them after the first dose, and then we moved them the Sinovac or the Sinopharm. But then there were other issues.
Dallin?
Lovely talk. I just wondered about the omics analysis you've done. Do you think it's possible to get that into the clinic and use it in a predictive capacity for outcome?
Okay, so to that, I think it's a very important question. Now, we were able to generate very good laboratory data, but actually some of the clinical data then it's a little challenging. So I think that is I actually believe that there have been some studies that attempted to do this, but I think the challenge is some of the incomplete data sets that make it very challenging.
Also wondering if the hallmarks you see the biomarkers are indicative of COVID specifically, of coronavirus infection in general,
Okay.
or respiratory infection?
Okay, I can share the paper with you. We did some comparison. We compared with the latest, the most recent respiratory infection, which was the influenza outbreaks in 2009. There were some similarities, like IL-6, which was why it gave the clinicians to try the Acoxia treatment. But there were some unique signatures, but some of this also could be due to the severity of the inflammation. So whether is it exactly specific to SARS-CoV-2, I think we need really a big data set before we could look at that, with different cohorts and all that.
Thank you very much,
Okay,
Lisa,
thank
for
you.
a great talk. Our next speaker is Roslin Alexander from University of Bristol, who's going to talk to us about aerosol transmission. Do you want to use the handheld?
Yeah, can do.
Is it turned on?
Hello? Oh,
Yeah.
There we go. It's like karaoke. Let's get this going.
5 minutes, Rob.
Great, thanks. Okay, so this is a collaborative project between School of Cellular Molecular Medicine, the School of Chemistry, and the Bristol Vet School, where we get up to our scientific capers, and we look at airborne disease transmission. As we know, the pathogens responsible for respiratory disease propagate between hosts on the aerosol droplets and particulates that we expire when we breathe and talk and cough. But there's 4 main areas which contribute to a successful disease transmission. The first one, particle generation. So where the particles originate in the respiratory tract will have an effect on the pathogen load, but then also the particle size. And that affects the movement around the room. So we know the big ballistic droplets will fall out of the air quite rapidly. They're governed by gravity, whereas smaller particulates will float around the room on air currents, a bit like cigarette smoke. The third one is infective dose. So how does the right pathogen reach the right tissue with the right amount to cause disease? And what we look at in our group is the pathogen decay in those droplets. So how long does the virus remain viable in the airborne phase, and does the composition of those droplets affect that? And this has a downstream effect on the infective dose. So we use an instrument called Celebs, and it's a electrodynamic balance. So we trap droplets between 2 electrodes, and there's viruses within those droplets. We've got tight control over the temperature and the humidity, and after intended incubation, we can release those droplets into the collection media and then assess the downstream infectivity with this assay. And this is the kind of data that we get. So this is infectivity against time, and this is at 90% RH. So this is a very humid environment. And this is comparing MHV, mouse hepatitis virus, which is coronavirus. It's a surrogate for SARS CoV-2, with my colleagues Henry Oswin's SARS CoV-2 data. And you can see at this high relative humidity, there's a gradual loss of infectivity over 5 minutes. So we expect these droplets to evaporate. They lose water and solvents and CO2 to the air, and these droplets would equilibrate as a smaller droplet. So within those smaller droplets, there's non-physiological conditions. It's higher solute concentration and increased pH, and that's what's driving this loss of infectivity. When we compare that with a more dry environment, 40% RH, this is like a typical office space. There's a rapid loss of infectivity that we see within the first 5 seconds of particle generation. And that infectivity remains pretty constant over the 5 minutes. And these droplets will dry completely into particulates, which are salt crystals and organic matter. And this process is called efflorescence, and that efflorescence process is driving this rapid loss of infectivity. But yeah, so MHV is an appropriate surrogate for SARS CoV-2. One thing I'll just say, these droplets are growth media. It's what we harvest the virus in. So it's a kind of it's a surrogate for respiratory fluid, but it's not the whole picture. There's more complex things in saliva and respiratory fluid. One of those proposed was mucin, so the building blocks of mucus. So does that have an effect on the infectivity in the airborne phase? So we just added clinically relevant concentrations of mucin to the droplets. And as you can see, with increased concentrations of mucin, there is a reduced loss of infectivity. So that the mucin is kind of protecting the virus from the damage from the efflorescence. And we see this kind of after 2 minutes, the same trend, but we don't see that after 5 minutes. So this is a transient process. So the theory is that the virus is being protected by the mucin, maybe coating or seconding the virus, protecting it from that loss of infectivity that we see at RH, 40% RH. Well, that was all MHV, and my colleagues have been looking at the different variants and their kind of stability in the air. So what we see is there's identical decay for these two variants, original and delta, at 40% RH. So that mechanism is the same for all variants. And after 40 minutes, there's 95% loss in the aerosol phase. That's independent of RH, so they catch each other up. So there's kind of two mechanisms of decay in this curve. But what we did see was the difference in decay between the variants at higher RH. So the original variant is probably more resilient to the non-physiological conditions in that evaporating droplet. Yeah, thanks.
Any questions, Rob? There's one at the back, James.
Yeah. Just to agree with you on that one. So do I have to repeat the question?
No, it's all right. I think we'll pass. It was whether or not you focus on the loss down to 95% after many minutes, or whether you worry about the fact that it's 95% infectious after a few minutes.
Yeah. So these, if I just go back to this slide real quick. So even after 2 minutes, there's quite considerable infectivity. And if you think how far a particle can travel in those 2 minutes, it's pretty scary. And kind of when we're talking about prevention and stuff like that, I remember during the pandemic, they had to have room changes between operations and things like that. So we're really focusing on the close proximity transmission here. And I think that's what's important, right? Thank you.
Okay, Jules, quick question. Jules has asked a very contentious question over: would mask wearing select for a variant that can pass through a mask?
So I think, to be non-controversial, I think masks are very good at stopping the virus transmission if they're properly worn.
Good man.
But I'm not I'm not sure if you could select with the masks. Yeah, I'm not sure about that.
It's an interesting question, but it's very definitely gain of function if you test it in the lab. Right, we should move on.
Thanks.
Thank you very much, Rob. Isobel. Introduction, I failed there. Isobel is going to talk to us about IBV, I'd imagine, and she's from the Pirbright Institute.
So I'm going to talk about some work about the cell type dependent effect of the T16A or A26F mutation in the E protein of IBV. So IBV is a gamma coronavirus which infects chickens, and I've used the strain BOR in this work, which is a non-pathogenic lab strain, because it has extended cell tropism, which has allowed me to use continuous mammalian cell lines. So I work on the envelope protein, which forms an ion channel during infection, and it's been reported that mutating two residues to generate either a T16A or A26F mutation abolishes this ion channel activity. So I've investigated this by generating these mutations in BOR, and I carried out NGS sequencing and found that the T16A or A26F mutation were present at consensus level. I then passaged the recombinants and found that BOR T16A generated revertant mutations in primary chick kidney cells in all isolates at passage 10, but in eggs, it generated revertant mutations at passage 1, showing that there's different selective pressure between the two cell systems. Interestingly, as well, the BOR A26F mutation was more stable, as only one isolate generated a revertant mutation at passage 10, and no reversion was seen in eggs. So I then looked at the replication kinetics and found that by and large, BOR T16A replicates comparably to the parental BOR. But in contrast, BOR A26F replicated comparably in CK cells, which is a chicken cell line, but not in vero cells or in eggs, which shows that there's both a mutation dependent effect and also that there's a cell type dependent effect in BOR A26F. I also looked at the cytopathogenicity caused by the recombinants and found that in the primary chick kidney cells, all the recombinants cause comparable cytopathogenicity to the parental virus. But in veros, the recombinant viruses were less cytopathogenic, which shows that there may be different factors involved in this in the different cell types. So overall, I've generated the T16A and A26F mutations in the E protein of IBV, and I found that the T16 and A26 residues appear to have different effects, and also that these effects are cell type dependent. And overall, this work highlights the importance of cell type selection when investigating this protein. So I'd like to thank everyone at the Pirbright Institute and University of Bristol who's helped with this work, and my funders. Thanks.
Questions for Isobel? I might have missed it at the start. Why did you choose these mutations?
They've previously been shown to inactivate the ion channel. So I wanted to see what effect inactivating the ion channel had on the overall virus in the context of a recombinant.
I'm going to show my background as a flu virologist here. I don't know what the IBV ion channel does. Is it entry or is it budding? Or?
It's assembly.
Okay. So is this saying then that there might be different budding mechanisms in different cell types?
Potentially, yes, or it has a lot of different interacting partners, so potentially there are different factors present in the different cell types. which affect the E protein and the characterization of it.
Questions from the floor? So the hand at the back first.
No, I haven't ever made one with both mutations, no, but that would be interesting. But I guess because they're both supposed to inactivate the ion channel, one should be sufficient to do that. So.
Ed, were you going to ask something? Okay, let's thank all our speakers for great talks, and